Thank you. I'm really happy to be here, and I'll talk about how GPCR recognize peptide ligands using machine learning and discriminant equations. And more, I share some new findings from this approach. So here's the quick outline of my presentation. First, I will discuss the non-peptide and GPCR interactions and our previously developed prediction methods to predict its interaction. And then I will introduce the new method named IDL score. And then I will break down the performance evaluation and newly identified the determinants in 3 steps. Let's get started with the peptide GPCR interactions. This is the phylogenetic tree of the GPCRs. Usually, the peptides and GPCR relationship is correlated with the sequence similarities. So, for example, neuropeptide Y interact with the five similar GPCRs named NPYR1 to 5. And more, the tachykinins interact with NK1R, NKR1 to 3, respectively. But substance P interact with the evolutionary unrelated GPCR named MRGPRX2. And more, the MRGPRX1 is the homologous gene of the MRGPRX2, but it interacts with the BAM822. It's the way dissimilar sequences are. So, the interactions of the peptide and GPCR cannot be described only with the sequence similarities. So next, so to predict its interactions, I developed the machine learning method to predict its interactions. First, I collect the interaction information from database and literature, and then I converted the amino acid sequence of GPCRs and peptides into our original descriptors. I will introduce these descriptors in next slide. Then build a trained model using the linear SVM. Then predict the new peptide GPCR interactions for unknown pairs. Using this method, we already found 11 novel peptide GPCR interactions without the evolutionary relationship. So then I move to the descriptor constructions. So usually GPCRs interact with the peptide in the extracellular domain of the alpha binding pocket surrounded by the 7 alpha helices. So usually, I extract the transmembrane region by global alignment. And theoretically, these alpha helices were aligned without gaps. So this alignment had the equal length. Then I assigned numerical values to each amino acid using the Z-scale vectors. This Z-scale vectors was the PCA calculated using PCA of the physical properties of each amino acid, and then convert the amino acid into 5 lengths vectors like this. And the important in here is the position of the each residue correspond to the vector components. And then I developed the peptide descriptors. Usually, experimental biologists often use the motif to classify peptide families like this, like this. We represented peptide as a bit vectors that shows whether or not it has a motif or not. Like one represents it has the motif, and 0 represents it has no motif. And this motif contains the single amino acid or amino acid categories defined in the profit descriptors. And then we represent the CPI pair descriptors as the cross product of the peptide descriptors Lx and the GPCR descriptors Rx. These descriptors were inputted to the linear SVM, and the discriminant of the GPCR and peptides were shown like this. Here, the AA to XX means the weight vectors run in the SVM. And each term in here corresponds to the peptide and amino acid pairs in amino acid residue pairs, like this. So this term represents these two GPCR residues and peptide residues. So it means we can show how much specific peptide GPCR residue pair contribute to the interactions. We call this like by extracting the terms, including the amino acid pairs. So we define the IDL score like the extracting by extracting these terms. And I visualize the ideal scores in the heat map like this. Red line indicates the high ideal score. It means that these residue pairs contribute to the interaction, and the blue lines contributes to the non-interaction. For performance evaluation, we look at the mutations that degrees interactions and unrelated activity for 5G PCRs. And I compare the prediction performance with the alpha missense, which predict the mutations pathogenicity. So as shown in here, pink line, our ideal score method showed the higher prediction performance for 5 GPCR peptide pairs than that of alpha missense. Notably, the high prediction accuracy for MC4R and CCKBR, which recognize various range of ligands, suggests that the ideal score can accurately predict important residues for different peptide structures. Next, we estimated the determinants of ligand specificity of MRGX1 and 2 discussed in the introductory section. So the BAM 822 interact with the MRGPRX1, and the SP interact with MRGPRX2. And then we estimated the determinant amino acid in each GPCR and exchanged the residues and assessed the activity using the intracellular calcium assays. This is the result of the BAM 822 ideal score analysis. This upper panel shows the MRGX1 and the BAM interaction ideal score, and the lower panel indicates the MRGX2 and BAM interaction ideal scores. And in here, L1.35 and S1.43 were MRGPRX1 specific high ideal scored residues with BAM 822. So we changed the MRGX1 residues of these two residues into MRGX2 types. As please see the panel C. As a result, S1.43A mutant of MRGX1 showed similar activity to the wild type MRGX1, but L1.35P MRGPRX1 showed the lower activity than the wild type MRGPRX1. It means that L1.35 is the determinant residues for the MRGPRX1 and BAM interaction. And also, we introduced this MRGPRX1 type mutation into MRGPRX2. As a result, normal wild type MRGPRX2 has no activity to the BAM 822, but the A1.43S mutant of MRGPRX2 showed the small response to the BAM 822. So this this result indicates that L1.35 is the determinant residue for BAM interaction. And similarly, we also similar analysis to the SP and MRGX2 interaction. In here, P1.35 and F3.24 and G4.61 were MRGPRX2 specific high ideal scored residues with substance P, SP. And the panel C shows this assay of the assay result. P1.35L mutation of the MRGPRX2 showed the similar activity to MRGPRX2 wild type, but F3.24I and G4.61W mutation showed no response to SP. And then we introduced the similar mutation to MRGPRX1. So wild type MRGPRX1 had no response to the substance P, but I3.24F and W4.61Z MRGPRX1 that mutant responded at 40% of the wild type MRGX2. So this result indicates that these two residues were determinant residues for the substance P recognition. And then we did the molecular phylogenetic tree analysis for the MRGPRX1 and 2. And the right panel shows the 1.35, the BAM 822 determinant residue, and 3.24 and 4.61, the SP determinant residues. Shown in this, MRGPRX2 has the conserved the F3.24 and Z.61 residues importance for the interaction with SP. This suggests that MRGPRX2 appeared in the common ancestors of Eurocont and gained the ability to bind SP. And then MRGPRX1 showed the concept the L1.35, but in some species, Evolutionary, L1.35 was mutated with 1.35. So this MRGPRX1 is seems to be lost the interaction with the BAM822. And more, we explored the L1.35 in MRGPRX2. So as a result, some primates had the L1.35 in MRGPRX2. So this MRGPRX2 seems to be the dual receptor for the SP and BAM822. And more, in another SP receptor of the mouse, MRGPRP2, F3.24 and G4.61 were not conserved. That it indicates the existence of the another molecular mechanism for SP recognition. So this is a conclusion, but it's extended time. Thank you for listening.